Welcome everybody. Thank you all for being here as we continue our monthly webinar series. This webinar is being recorded, and afterwards the recording will be uploaded to our YouTube channel. This webinar is being hosted by the Arkansas Native Plant Society. We've been around since 1980. Our mission is to promote the preservation, conservation, study, and enjoyment of native plants in Arkansas, and to educate the public on the value of native plants and their habitat. If you'd like to learn more about the Arkansas Native Plant Society, you can do so by visiting our website at anps.org. You can check us out on Facebook at facebook.com/Arkansas Native Plant Society. And I'd like to encourage you to join the Arkansas Native Plant Society. You can do that on our website with a PayPal account. Go to anps.org/join. Another option is to mail a check in to our treasurer, which her address is there listed there on our website. On that page, amps.org/join. I want to give a quick plug for the next two webinars we have coming up in our series in August. On August 4th at 1 o'clock, that's a Wednesday, Dr. Subedi will be giving a talk on oak diversity in Arkansas and biogeography. And then our September webinar will be given by Dr. Richard Abbott, who will be talking about native vines of Arkansas. Our speaker today is Dr. Adam Schneider. Adam is an assistant professor of biology at Hendrix College and the curator of the Hendrix College herbarium. He completed his Ph.D. at the University of California, Berkeley on the taxonomy, systematics, and genomic evolution of parasitic Orobanchaceae. Since moving to Arkansas in 2018, Adam teaches a variety of courses in botany and environmental studies and maintains an active research lab. Today, Adam, or Dr. Schneider, I should say, is going to be presenting on the parasitic plants of Arkansas. So with that, I'll hand it over to Dr. Schneider. Thank you.
Great. Thank you so much, Eric, and thank you everyone for tuning in. I was just saying before we got started, as a recent transplant to Arkansas here, I've been I'm excited to be able to participate in this Native Plant Society group, and I haven't really had the chance lately with COVID and some other things, so hopefully I'll get the chance to meet many of you in person before too long. Today I'll be talking about the parasitic plants of Arkansas, and you can see right here representatives of five main groups of parasitic plants here in Arkansas. So just a quick little outline before I get started. I'll probably spend the first maybe 15 to 20 minutes just talking about what is a parasitic plant. There's certain misconceptions and just how do parasitic plants function and how are they different from other types of plants. And then I'm going to talk about two major groups of parasitic plants, or two different ways that plants can be parasitic. One of those is called Mycoheterotrophy. I know that's a bit of a mouthful, but I'll explain what all that means in a little bit. And then I'll also be talking about some of our parasitic plants that actually form direct connections with other plants. These are called the hostorial parasites. So you can see some examples. These are the 5 different examples of parasitic plants that we have here in Arkansas. These all represent independent evolutionary origins. And so you might think that all parasitic plants are each other's closest relatives, but that's not true. So they're similar in a lot of ways, but also different in some unique ways. And I'll be walking through each of these 5 different groups to talk a little bit maybe about how you identify them. But I also want to focus a little bit on their ecology and evolution and natural history. And so I'll be integrating certain research studies that touch on those different themes with these different groups of plants. So I'll just get started with a sort of a simple definition. Well, what is a parasitic plant? And when I'm talking about parasitic plants in this talk, I'm referring to plants that get their water, nutrients, or sugars or energy from the living cells of other organisms. Certain people might have misconceptions or will often think about, well, what about resurrection fern? Is that a type of parasitic plant? And for the purposes of this talk, I'm not going to consider any epiphytes. So epiphytes are plants that just grow on the surface of other plants, like these beautiful ferns, but they're really not, taking their resources from the living part of that tree that they're on or anything like that. And so they're a lot more ecologically distinct or physiologically distinct from from the other organisms. So we won't be talking about that in this talk. Similarly, there's lots of plants that are vines. You might have a chance to hear about that in a couple months. But often can kind of look parasitic, right? Climbing up other organisms and covering them sometimes quite densely, even to the extent that they'll kill or suffocate the plant that they're growing on. Here's poison ivy as an example, but you can think of other types of vining plants, or what I might call structural parasites. So that in these cases, they are reliant on their host plant for stability, or for mechanical support. But again, they're not getting any of these resources I talked about before through their host. And so again, I'll kind of leave that aside and not talk about that during this presentation. So the first question then is, well, how do they get these resources? How do parasitic plants parasitize? And many of them, not all, but many use a special organ called a haustorium. And so you can see an example here of a Dodder plant. I'll be talking about these a little bit more later on. These, you may be familiar with these from seeing them out in Arkansas. These orange kind of twining plants, Dodder, or the genus Cuscuta, will form direct connections with a special organ called a haustorium. So you can see this really clearly in the picture on the upper left, where the parasite is on the outside and the host stem is below. You can see the host stem has kind of bigger cells, kind of has that angular shape. And then the parasite is right on top. And you can see that there's a part of the parasite that's actually growing into the tissue of the host. And that's what the haustorium is. The job of this part of the plant is to attach to the host tissue, specifically the vascular tissue generally, and siphon off those resources that are important. So in this case, you have a direct cell-to-cell connection between the host plant and the parasitic plant. Now, haustorium can attach to the stems or the roots of different hosts. And this is going to vary depending on the type of parasitic plant. So Dodder, for example, is only going to attach to the stem of their host, but there are other types of parasitic plants that will form haustorium that attach to the root of their host. And you can see an illustration of that on the two images that just popped up on the right. The lower image shows the darker colored host root along the bottom and the lighter colored parasite root above. And you can see it's connected, in this case, it's connected in 2, maybe 3 places with a large kind of swelling of the parasite root near where it's attached to the host. If you're interested in seeing that on a cellular level, again, I included this diagram or this image on the upper right. And this is a cross section again. So someone has sliced through the root. The H on there stands for host. So that's part of the host tissue. And you can see this little kind of kinked structure that's inserted itself into the host root that has a P on it, P for parasite. And that there again, you have the parasite growing into the root of the host. And that interface between the host and the parasite is illustrated by these black arrows. So this is a very close connection physiologically. And you actually see not just inorganic molecules, or not just inorganic molecules and sugars, but also more complex biological compounds being transferred from the host to the parasite. And I'll talk about that a little bit more later. So those are the haustorial parasites. Now, another group of parasites that I already mentioned are called the Mycoheterotrophs. So let's just kind of break down that word for a moment. Hetero meaning other, and trophy refers to how the plant or organism gets its resources. So in this case, these plants are not making their own sugars through photosynthesis, but they're getting it from another organism. In this case, that other organism are symbiotic fungi that live in the soil. And that's where the myco part comes in. So mycoheterotrophs are plants that parasitize fungi that have a symbiotic relationship with a green free-living plant. So for those of you who haven't maybe thought about this as much, it can be a little bit complicated, and it is, sort of a complex ecological relationship, which is what makes these plants so interesting. So here's a diagram on the lower left that you can kind of see, what's going on here. So many plants, especially woody plants, benefit highly from symbiotic relationships with fungi in the soil. And so these are, symbiotic fungi or mycorrhizal fungi. And so these the dark green lines you can see are the fungal hyphae, or the subterranean cells, or body cells of the fungus. And they'll connect with, different trees and other roots in this really important interaction, where the fungi is helping the tree or plant gain nutrients, maybe by weathering them out of the rock or inorganic parts of the soil, and also by absorbing water and other resources, from the soil. In exchange, the photosynthates, so the sugars and energy that's produced and captured by the tree, is then shared with the fungi. So in this case, it's a win-win ecological interaction, right? But what's interesting about these parasitic plants is they're parasitizing the fungi that is then getting its sugars from these, these photosynthetic trees nearby. And it took people a long time to figure out that that's what was going on here. You can see this is just a table that I pulled out of a paper talking about different researchers in the 19th and 20th centuries that were trying to figure out how these plants can survive, without being able to do photosynthesis. So Monotropa is a genus of plants that's found in Arkansas. I'll be talking about them a little bit more later. But ghost pipes or pine sap, if you're familiar with those common names, that's Monotropa. And you can just see by looking at this table, first of all, that it's not necessarily a linear process, of a better and better understanding. People disagreed, sometimes, these scientists would make observations that turned out not to be supported and so forth. But by the middle part of the 20th century, we had a pretty good idea of, how this is happening. And in fact, the fungi are able to, or they must penetrate the cells of the Monotropa. So, one model for how these plants are able to recruit their fungi that they end up parasitizing is, by acting like they're going to be this, this nice partner, right? Like the fungi is used to with other plants. But then, once they form that connection, these parasitic plants, don't really have anything to give. So anyway, there's a lot of research that has gone into, these things that maybe seem pretty cut and dry today. Now, moving on, another kind of bullet point to keep in mind is that plants can both be fully parasitic, and these are the ones I'm going to be focusing on today. All of the plants I've shown here on these slides are Arkansas native plants. And I think the ones that are most unique are the ones that are fully parasitic, because they've given up, the ability to produce, to do photosynthesis, which is, I think, is one of the first things that people associate with plants. So we have coral root orchids, we have the cancer root, the genus Afilon, we have the Monotropa that I just talked about previously, so you can see some pictures of those. However, in many instances, there's actually plants that you may have seen out and about and never really thought about them as being parasitic, because how would you know? This Aegolinus, for example, it's perfectly capable of photosynthesizing. It's got green tissue, leaves. For all intents and purposes, it looks like a normal plant. But in fact, this Aegolinus here is a relative of Afilon. They're in the same plant family. And so you see this whole spectrum of ecological interactions that in the case of these partially parasitic plants are quite a bit more obscure. Mistletoe is another one that's maybe a little bit more obvious that that one's a parasite despite its green foliage. And so if you're interested in evolutionary biology and how these plants came to be, you see this really interesting spectrum of parasitism. And so you can range from facultative parasitism, where plants like this Indian paintbrush are perfectly capable of persisting on its own. In fact, one study showed that about 2 of the 4 species that they looked at could grow perfectly well without parasitizing. But yet, when they were able to form root connections with other plants, they would grow 3 to 41 times the size. And then on the other end of that spectrum, you see complete loss of photosynthesis and obligate parasites like this Afilon. So all of this together leads to this spectrum of traits that some people have called parasite reduction syndrome. And you'll probably notice as I start showing you some of these native plants in Arkansas that they show this convergence in morphology. A lot of the different parasites have a shorter stature, loss of leaves, stems, roots, other features that most other plants have. But in this case, it makes sense, right? Why do all the work of putting out leaves when you can't use them, and you can get your resources another way? And they also tend to have very small seeds. This plant on the upper right is a relative of mistletoe that's actually not found in Arkansas. This is a dwarf mistletoe. So if you go out west or into the Caribbean or the northern part of North America, you'll see them. They're even more reduced than the mistletoes that we see, right? So these little orange, yellow-orange structures coming right out of the stem of this spruce is that dwarf mistletoe. You see physiological changes, loss of chlorophyll, and changes in function, even of the plants that are still able to photosynthesize. And you see some pretty remarkable genomic changes. I won't talk about that here. It's some previous research that I've worked on. What this is just this image that popped up in the middle is just a map of the chloroplast genome of some of these plants. And you see losses of many different genes and a reduction in the overall size. Finally, before I'll talk about our native plants, I just want to put this into their global context. How many parasitic plants are there? For the hostorial parasites, it's pretty easy to count, right? You can just do excavations, look at their morphology, and see, oh, yep, look, there's a hostorium. There, it's attached to this other plant. And about 1 to 2% of flowering plant species are hostorial parasites. And that's almost about 5,000 species at the last count. What you're seeing on the right here is a cladogram. And so if you're not familiar with these diagrams, they're basically like family trees of different plant species. And so these tips that are kind of in the center are all sorts of different plant orders. An order is just a taxonomic group that is more inclusive than the family level. And so this shows all the flowering plants, and groups that are highlighted in red are groups that have these independent origins of hostorial parasites. And so you can see they're pretty well distributed, lots of diversity there. In Arkansas, we have 3 representatives of that: the Orobanchaceae, the Dodders in the Convalvulaceae, and then the mistletoes in the Santalales group. The Mycoheterotrophs are it's a lot more difficult to get a good count of that. Part of the reason is because there are many completely autotrophic plants that have important mutualistic relationships with fungi. So you can't just look and see, oh, are there fungi associated with this plant or not? And so that's why this particular count only looked at full mycoheterotrophs. So ones like those coral root orchids I was showing, or the ghost pipe that have completely lost photosynthesis, right? If they can't photosynthesize, you know they can't live on their own. And so then you can get a better count. And this way, and by this count, they've documented at least 500 species that are full mycoheterotrophs, many different evolutionary origins, but mostly monocots. Most of the diversity here is in the orchids. All right, so let's look at some of Arkansas's native plants, and we'll start right here with the mycoheterotrophic orchids. There are 3 different species distributed across 2 different genera in Arkansas. So I have pictures of each of them, and these maps, which I copied from iNaturalist. If you're not familiar with iNaturalist, it's a really great tool for documenting biodiversity. Learning about biodiversity, you can submit images of different things that you see while you're out and about, and you can identify them. Other people can identify them. It's really cool tool. I know we have the of course the very good atlas of native plants in Arkansas with all the different maps, range maps. I opted to use these because the number of points that you see, these iNaturalist observations, can also give you a relative idea of how abundant or how easy they are to find. How likely are you to to stumble on this? So again, these 2 different genera, Hexalectris spicata, and then 2 species of Corallarhiza. You can see generally these are found in the Ozarks or Ouachitas, and there's a few populations up on Crowley's Ridge. And like the other microheterotrophs, you'll just see these these spikes, these inflorescences sticking right up out of the ground. So like that parasite reduction syndrome I was talking about, not really appreciably sized leaves. If you do look closely, you'll see small little scale-like leaves. But this is it. If you're going to see these plants, this is what they look like above ground when they're in flower. And so as a result of that, there's also a strong seasonality in terms of when you see, or in terms of when these plants are out. And that's one of the ways that you can tell the 2 species of Corallarhiza apart. Right, one of them is more spring to early summer flowering, and then the other is later in the autumn. A couple other features that you might think about too is looking at the overall size of the flower. Hexalectris has generally larger flowers, and they'll have real conspicuous bracts under their inflorescence bracts, whereas in Corallarhiza it's much less conspicuous. You can see that image of Corallarhiza odontorhiza with the smallest flowers, and then very small bracts there. The Corallarhiza has quite a bit of other diversity in other states and places as well. But as far as we know, there's just these 2 species here in Arkansas. Let me talk a little bit more about the life history of these plants in particular. This is a study done, I think, on a slightly different species, but the big picture is the same. One of the challenges of these parasitic plants is, or root parasites at least, is trying to figure out, are they annuals or perennials or what? Because a lot of these plants will live for several years underground, before emerging from the surface. So what you can see here in this upper left, when the seeds germinate, they'll develop into these protocorms. So a corm is just a modified stem that is, helpful or useful in either storage of resources and or clonal reproduction. And so you can see that starting to develop here in this panel number 1. And it'll just sit and, acquire resources for several years. In this case, this flowering shoot on panel number 2 has been 4 years old already. It takes the plant that much time to just be able to put up these big flowers or inflorescences. And then at that point, the underground rhizome, once it flowers and sets seed, the rhizome will senesce, and you'll get these little tiny pups coming out. These, part of the rhizome will die, and then you'll have the other parts that are now kind of separated and individual. And you can also see that here on panel number 4, you can see a whole range of new buds that could then start growing again clonally on the darkened kind of senesced rhizome. And so this is why you'll often see them, if I go back, in these little clusters, right? So this is all probably one clonally expanded group of stems. One thing that's not shown in these above images is that the fungi that's associated with this Corallarhiza or any of these parasitic orchids. And that's what you can see here on the bottom. This is a cross section of the root. On the left is a drawing to help you orient yourself. On the right is an actual photograph of that taken through a microscope. And these dark lines look like almost striations. Those are the fungal hyphae cells that have surrounded the rhizome of these plants. And then in some places, which I think are indicated by these little wedges in the image, you actually have the hyphae growing into individual root cells of the plant. Like I said, these orchids will recruit the fungi, but instead of a nice symbiotic exchange of resources, it's mostly an exploitative relationship here. The next group of plants look somewhat similar superficially, but are in the family Ericaceae. So Ericaceae includes things like vaccinium or the blueberries, huckleberries, and so forth. It includes rhododendrons, azaleas. So and it includes these guys, this genus Monotropa. We've got 2 species here in Arkansas, Monotropa hypophytes, they're the pine sap, and Monotropa uniflora, the ghost pipe. And some ways to identify these in the field, they do have some phenological differences. So that's one, the timing of when they're out and above ground. But you can also see there's a difference in color that's usually pretty consistent. This pine sap is more of a yellowish color, yellowish red, whereas the Monotropa uniflora, as the name implies, is generally white, sometimes tinged pink like this. Also, the number of flowers on a stem is a good way to distinguish them, especially if the plants are older or in fruit or whatever, some other stage of their life cycle. As the name implies, uniflora, 1 flower per stem for the Monotropa, whereas on the pine sap, you've got multiple flowers per stem, as you can see here on the left. When they come out of the ground, the whole inflorescence will be nodding like this, as you can see in most of these images. And then as it becomes fruit, as it matures into fruit, the whole, each of the flowers become erect. And you can see that clearly on this photograph on the left. One thing that's kind of cool about these mycoheterotrophs is that people have done studies to see which types of fungi different species associate with. And it turns out that these 2 species will associate with completely unique families of fungi. And so as a result, you'll also probably find them in slightly different habitats, or you often can find them in slightly different habitats. And I'll come back to this a little bit later in talking about the role of host specificity in kind of understanding the ecology of these parasitic plants. And these are the only two species of Ericaceae or Ericaceae Mycoheterotrophs we've got in Arkansas. So we're up to five species now, 3 orchids and 2 of these guys. Now, I'm going to shift gears now to starting to talk about some of our different hostorial parasites. And just as a reminder, these are the ones that form direct cell-to-cell connections with host tissue, specifically vascular tissue inside the plant host, inside the host plant. So first, I'm going to start with the mistletoes. And we have one species of mistletoe here in Arkansas. This is probably the parasitic plant that people see the most, especially in the wintertime. You know, it's pretty apparent in these deciduous oaks and other trees that often will you know, host these mistletoes. So there's a lot of pictures here. Probably should have added them one by one, but first I'll start with this range map in the lower right. As you can kind of see here, it's pretty evenly distributed around the state. You know, often can be found in urban areas as well. And in terms of identification, again, very easy. This kind of shrubby clump or small shrub that forms inside the branches of other trees has these, these green kind of paddle-shaped leaves. And then if you've ever seen it in fruit, you'll see this kind of sticky white fruit. And you can see that, in this image on the upper right. And then sometimes, when the fruit falls off, you'll just see these kind of bare stems with the receptacles or the places where the fruit used to attach still on there. These plants have a really interesting dispersal strategy. You can see how sticky they are on this little inset image with this, on the fruit coating. And this is going to be really important, right? If you're a mistletoe and you want to live up in the branches of trees, you don't, how are you going to keep from spreading your seeds and just dropping them to the ground? And as parasitic plants, as obligate parasites, these organisms can't survive without a host. And so, so what do? Well, first of all, you want to be able to stick to the trees. And second of all, maybe if you're nice and fleshy, you can attract some birds to help move you from tree to tree or organism to organism. And I'll talk a little bit more about that in just a moment. In terms of the life cycle of these plants, on the upper left, you can see a mistletoe seed. This is actually a different species of mistletoe, which is why the seed or fruit maybe looks a little bit different than the picture on the upper right. And you can see it's just germinated, and the first thing it does is stick out these, these 2 little hostoria to then bore into and grow inside of the stem of its host. Here, but right beneath that, you can see a longitudinal section of a branch, a woody tree branch, and you can see that part of the mistletoe is growing inside of the wood of that plant. The dwarf mistletoes that I saw, or that I showed you earlier on, that aren't in Arkansas but are found throughout North America, those are an even more reduced version of these phorhodendron that can produce systemic infections. And so they have enough of their body inside their host plant that as the tree continues to grow, it'll just the parasite will just get carried along with it and will remain infected as the plant will remain infected even as it puts on new growth. Eventually they'll grow up and they'll flower, and these mistletoes have These are called dioecious plants. And that means that there's separate individuals that produce only pollen or only seed. And so that's, people often will colloquially call that male versus female flowers. So next time you see a mistletoe in flower, you can pick off a branch, and if you look really carefully, you can see, is it producing pollen, which will look more like this image above, or is it producing, or will it produce seed? Now, a lot of people, in general, parasitic plants, you think, oh my, these are going to cause all sorts of problems and to their host. That's a question that I get a lot. And maybe it's a little bit, my bias is just somebody who studies these organisms and just really loves them. But for the most part, they're not a huge threat to the host. For these mistletoes, for some of the other parasites that will only attach to a single host, that kind of makes sense in that, if you're so detrimental to your host that the host dies, then you're out of luck as well. So most of the time, a baseline mistletoe infection isn't going to be too bad for trees, although it can really exacerbate drought stress. And so that's really when you see negative impacts on trees, is when they're already being drought stressed and they have this parasite infection. One of the ways that parasitic plants will can get all of these resources from their host is being really wasteful with water, so to speak. And so by doing that, imagine that these cells in the host and the parasite are these pipes that are all connected. And so the parasites essentially will spring a leak, in their pipes, and they'll be releasing all sorts of water into the atmosphere. And that's going to pull material along that includes nutrients and sugars and all the other goodies from the host plant. So, so when the tree's already being stressed, having the water loss more than anything else is going to be really bad for these mistletoes. There was one study that was done that I think found during normal times, the mistletoes maybe only cause mortality in about 4% of trees, that they were being parasitized in an area. But then during this severe drought, 2/3 of the plants that died because of the drought were heavily infected with the mistletoe. But on a more cheery note, we can talk about some ecological benefits to other organisms. So first of all, these mistletoes, the phorhodendron is the only host for this particular species of butterfly, the great purple hair streak. These images show an egg on the upper left, the caterpillar on the lower left, and then male and female adult morphologies on the right. But there can also be ecological benefits to the host, maybe not the host individual, but the host species at large. And so this is one of the stories I find most interesting, which was a study done about 20 years ago looking at mistletoes, junipers, and this bird named Townsend solitaires out west. So we'll start the story with this phorhodendron juniperanum. So this is a species that parasitizes juniper bushes in the western juniper trees in the western United States. You can see, here on the lower right, you can see an infection there. These kind of darker bunches are the parasite, the mistletoe on there. And so because being parasitized is a negative is a negative effect on the host, that's why I have this little minus sign. Just so you can kind of follow through. Because there's going to be, it's a complex story, but the take-home message is really cool and I think worth thinking about. So there's the solitaire, and this bird will eat the cones, the fleshy cones on the juniper, and of course that's a positive benefit to the host, to the juniper, because it doesn't eat all of them. Some of them it drops, and so it helps disperse the juniper tree. Well, the bird also disperses the mistletoe, right? So it's equally attracted to these blue juniper cones and the white sticky fruits of the mistletoe. And so, okay, well, so maybe that's another minus because it can help move the mistletoe around as well. But what's interesting is that if there's enough mistletoe fruit, so if you have enough mistletoe on there, you're actually going to see that it attracts birds overall. So that's what this bar graph shows on the lower left. Just that trees that were infected with mistletoe tended to have more solitaires visit them than trees that were mistletoe free. But the coolest, what I thought was the coolest part of this whole story is that they also showed that by attracting more birds, it actually helps the junipers disperse better. So figure 4 here is kind of the take-home message of this story, is that areas, stands of juniper that were infected by mistletoe showed about 2 to 3 times more juniper seedlings coming up than stands that didn't have mistletoes at all. And so in this system, some mistletoes can actually be a good thing for the juniper, right? If there's too much, sure, there's going to be negative effects overall, right? But if you have a medium amount, you'll actually see that three-way interaction between the host, the parasite, and these dispersing birds help provide a net benefit to seedling recruitment and dispersal of the junipers. So, it could be similar things happening here in Arkansas. I'm not sure. All right, we've got 2 more groups to go, and the next one, which is also pretty commonly observed, is the genus Cuscuta or Dodder. These guys I call morning glory's evil twin, because they're actually in the morning glory family. Like morning glories, they'll twine up their other plants. Unlike morning glories, they'll attach via stem haustoria and then parasitize their hosts. Now, just first mention the Cuscuta worldwide, because this is, this genus is found on nearly every continent. And there's approximately 200 species, but fewer than 10%, probably closer to about 5% of them are really agricultural pathogens. In a lot of different places, you'll see Cuscuta on state or, local noxious weed lists. And I think it's, we can look a little bit closer, and it's not all Cuscuta that are going to cause problems, even though they can, cover their hosts pretty severely, like this case in the upper right. The closest relatives to them is actually not known because they're so, we know that they're in the Convolvulaceae family, but because they have all these morphological changes and genomic changes as a result of their parasitic life history, scientists still haven't been able to figure out what their closest relative in the family is. And finally, identification of these guys can be really challenging. And there's a few reasons for this. Identification keys could be vague or laborious. And sometimes there's obvious differences between different species that don't really show up well on keys. And I'll show you an example of that in just a moment. And globally, many species are only known from the type specimen. The type specimen is kind of a reference or standard that is designated when a species is first described to science. But unfortunately, in the case of Cuscuta, there are lots of species where we only have that original species description and maybe the specimen that was cited. And so it's hard to get a good sense of, what kind of variation does exist in this species, where does it grow geographically, what's the extent of its range, and so forth. Most Cuscutas actually can do limited levels of photosynthesis, despite it doesn't really look green. for the most part, although some parts of the plant can tend green. But there are two clades, or you can just think of them as groups of Cuscuta that are non-photosynthetic. But neither of those groups are found in Arkansas. So here's what I wanted to illustrate about these differences in habit. So here's an example of a species of Cuscuta from Hawaii, Cuscuta sandwichiana, that has this really, it just looks like, I think it looks like Rapunzel, just this trailing, these golden stems. And this particular species lives on beaches or coastal areas where there's a lot of low-lying vegetation. And so you can keep low to the ground and find additional hosts to parasitize. Whereas look at the contrast with this other species, Cuscuta denticulata, which is just, it almost looks like it's sitting on top of this host plant like a hat. And these new stems are pulling, always ascending, always reaching upwards for their next host. And of course, these plants typically, this species typically parasitizes shrubs in the Western American deserts. So again, this is something, one of these field characters that's kind of hard to translate into a key. But let me give you a few identification tips if you're interested in that sort of thing. There's about 10 species in Arkansas and plus a variety. And first of all, you're going to need floral tissue to identify these plants. If you just have the twining stems, it's not going to work out. And so some of the features to look at are the calyx and the corolla shape. How large, well, the shape and also their size. How, are they pointed? Are they rounded? And so forth. And also there's this feature called infrastaminal scales, which it's still not entirely clear what their function is. You can see on this lower right image, in between the stamens of the flower, there's these little fringy appendages on the inside of the flower. And those can be really useful for telling apart different species, whether they're present or not, their shape, and other aspects of their morphology. It's not clear exactly what their function is, but the leading hypotheses have something to do with reproduction or the pollination biology. And finally, consider host affinity. When you see these plants, I'll just go back quickly, sprawled over an area or a landscape, it can be easy to just assume that they'll parasitize anything in their path. But a lot of the times that's not true. A lot of the times they'll be preferring certain hosts and leaving others alone. And those kinds of affinities can be helpful in identification. I'm a little bit short on time, so I'm not going to go through these one by one, but here are just 4 common species throughout Arkansas, and some of the ways that you might, some of the diagnostic features and photographs. This was all taken from a paper that's available online, open access. If you just follow that URL, it's about the Cuscuta of Alabama. But many of the species that are in Arkansas are also here in Alabama. And so you might find that a useful resource because it also has some really nice photographs of each of the different species. You might also check out, the recording of this talk. You could pause the slide or take a screenshot or something if you want to read through here more carefully. You also might follow this link, write that down or something. There's a cool time-lapse video that you can see some of the life history of these plants. But I'll just walk you through that quickly here with some still images. This is what the seedlings look like, or the seeds shortly after they've germinated. Just these small little stems ascending up from the ground. And the first thing that these plants have to do is they have to find their host, usually within about a week or so of germinating. And they'll do that through chemical volatile cues. So vaporized chemicals that are exuded or released from the host plant sometimes can be detected by these Cuscuta plants and trigger them to grow in that particular direction. But also these plants will also kind of grow like other sorts of vining plants. Sometimes we'll find, especially in the morning glory family, we'll find their something to hold on to just by kind of spinning around, right? And if they crash into something, they'll use those touch cues to more carefully wrap around that particular object. So here's an example of that. In this case, they've tricked the Cuscuta into trying to parasitize just this wooden dowel. You can see those haustoria starting to form. And just as an aside, sometimes the host or a potential host can resist the parasitism. In this case, that image on the upper right, the host did it by killing off all of its cells that were nearby the point of attachment of the Cuscuta. And oftentimes that can be effective in preventing an infection. And then finally, once these plants have developed at least one haustorium, they'll just continue to grow and form additional haustoria with other plants, other individuals, whatever hosts they can find and that are susceptible to them. And they can also regrow vegetatively from the haustorium. And you can see all sorts of new little sprouts coming out of that haustorium here in this image that is on the lower right. For this reason, it can be very difficult, Cuscuta can be very difficult to eradicate, for example, from your garden. If it pops up and is parasitizing plants, you really have to make sure to get rid of every little piece of it, or else it will regenerate. By the time the plants are mature like this, they don't have any roots, and so they've lost the parts of the plant that were just in contact with the soil have kind of withered away. They really get all the resources through these haustoria. I think in the interest of time, I'll just skim over this, but there's been really interesting ecological studies that have looked at how Dodders parasitizing multiple hosts can provide a biological connection for materials moving not just from the parasite to the host, but from one host through the parasite to another host. And so you see a lot of different features, nucleic acids, proteins, even pathogens can be transported through these Cuscuta stems. And so was going to walk through a study example of that, but looking at salt stress, but I think in the interest of time, I think I'll skip over that. I want to finish by talking about the Orobanchaceae, the family of plants that I've spent the most time in my research, specifically in this genus called Afilon, which formerly was known as, or it was subsumed within the genus Orobanchaceae. However, genetic research, and there's several different morphological and biogeographical differences too, support that our native North American species should be recognized in the separate genus Afilon, distinct from Eurasian relatives, which have retained the name Orobanchaceae. Also in Arkansas is our beech drops. In places where you have beech trees, you'll have these plants parasitizing the roots of the beech trees. And in addition to their differences in host, Afilon being typically on asteraceous perennials, solidago, goldenrods are a common host for them around here. You can tell these apart by the number of flowers per plant. And so these Afilons will only have 1 to 2 flowers per plant versus many flowers per plant in the epiphagus. Sometimes the Afilon will grow in some pretty dense clusters, and there might be a whole bunch of plants stacked up on top of each other. So don't get confused if you see a clump of them. Each individual is only going to have 1, maybe 2 flowers on it. Another really obvious difference is in Afilon uniflorum, all of the flowers are on these little stalks, these pedicels, whereas in Epiphagus, the flowers are directly attached to the central stem, and so they're sessile, or almost sessile. In both cases, these plants are less commonly seen. Again, these Afilons in particular are really delicate plants, and So when they're not flowering, or shortly after they flower, they will very quickly die back, release their seeds, and will not be visible above ground. There is a large group of relatives that are also parasitic plants that are the hemi-parasites, or the photosynthetic parasitic plants. And so I think I've listed all the genera here. I apologize if I've accidentally overlooked one. But all of these plants form or can parasitize other plants. They'll just do it underground, and usually it's less important to their life history than these Afilon plants are. But all of these plants are indeed parasitic. Just a quick run through of life history again. Very similar features in some ways to some of these other parasites. On the lower left, you can see this black, it looks like dirt or something along this dead flower stalk. Those are the seeds of the parasitic Orobanchaceae. They're very, very small and very numerous. The abundance helps them, get lucky that one of them might come into contact with the host. But because they're so small, they really need to attach to the host plant right away. And so they'll do that by growing towards chemical cues exuded by their host root. You can see that in a time-lapse series of photographs on the bottom here. Then we'll attach to the roots. So here are some pictures of developing haustoria in the Orobanchaceae. And this is all mediated by this type of plant hormone called strigolactones. And then finally, these will look similar to the photos you saw earlier, where you have these direct, first penetration of the host root, and then these direct cellular connections. So what you're seeing here in this image, and I just have to show this because I just think it's so cool. On the left is a parasite cell. On the right is a host plant cell. And these dark channels that you see in between are called plasmodesmata. And these are connections, intercellular connections between the host and the parasite cells to facilitate the transfer of materials. Similarly, here you can see a parasite cell growing into a little pore in the cell wall of one of the host cells. So, really insidious little critters. The P is the parasite and the H is the host cell. And I think and then finally, just a whole number of different host defenses, right? So part of the reason why you have such host specificity is that some plants are better than others at defending themselves against the invasion of these Orobanchaceae parasites, or other parasitic plants, the Dodders, the mistletoes, and so forth. And anything you can think of, I think somebody's found a plant that can defend itself that way, either through structural or physical barriers, or chemical barriers, or sending out hormones to mess with the physiology of the parasite. You see all sorts of cool stuff. And so finally, as they become a mature adult, you can see in these images, these aren't Arkansas native plants, but they're relatives from other species that are found in the United States. But they just really well show this root connection, right? So there's the parasite I've drawn purple boxes around, and everything that's attached to the base there is all the host root. And these Afilons and also Epiphagus as well. They can't send out roots and look for new hosts. They really have to attach early on in their life cycle, and just they're kind of one and done in terms of their host affinities. I don't think I have time to go into this in too much detail, but just that some of my own personal research into the Afilon has used phylogenetics or genetic relationships to show that there's a lot more diversity in species than we first thought. And so here's just one example of what used to be traditionally considered two species, Afilon uniflorum and Afilon fasciculatum in North America. But when you do the genetic analysis, you see all of this structure, this, so these long, branches of independent, genetic changes illustrate that there's really a lot more species than we've thought. And if we look even further, we'll see that each of those different lineages or cryptic species have their own host preference. So I guess the take-home message for here, maybe as a field biologist or as just an enthusiast, is pay attention to the host that these parasitic plants are on, and that can tell you a lot about not just what species you're looking at, but also its ecology, where you might expect to find others. And in this case, my research has shown that these plants are a lot more host specific than we had thought, because this isn't really for two species. There's actually at least 5 species here, mostly in the Western United States. The plants here in Arkansas got to keep the name Afilon uniflorum. So with that, I think I'm a couple minutes over, so I'll wrap up, and maybe there'll be time for questions. But if you don't have any questions now or tuning in, feel free to send me an email anytime. And just a quick thanks to some of the co-authors on the research that I presented of my own, but more importantly to all of the people who've made herbarium vouchers to help document this biodiversity, and also iNaturalist observations, including certain people whose iNaturalist images I shamelessly used in this talk. So thank you all for your attention and your interest, and we'll see if there's time for questions.
Yeah, thank you, Adam. Appreciate that. That was a wonderful presentation. And yes, we do have a couple of questions already. Just want to remind everyone, if you have questions, please type them into the chat box. John Barr asked a question. It was just before you started talking about the ecological benefits of mistletoe. But I think this question would still be relevant to the other native plant or the other native parasitic plants. So you had wondered, do you have any comments on pollinators for the various parasites?
Yeah, that's a really good question, and actually something I wish I knew more about. In Afilon, I guess I can speak to that kind of the best, is that there isn't really good knowledge about what kinds of things pollinate these plants. What's interesting in Afilon is that you see this wide range of floral shapes and colors that you might think would be attracting different types of pollinators and whatnot, but people just haven't done the basic natural history studies to really figure that out. Who's visiting these different plants, who's actually successfully pollinating them. Some plants, in the Orobanchaceae, sometimes you'll see what are called cleistogamous flowers. And these are flowers that actually will not open, but self-pollinate. The plant will self-pollinate itself. So the flower never opens and then the seeds develop. And you'll often see both cleistogamous flowers and these big open flowers that are called chasmogamous flowers on the same individuals. So some of these plants might be doing kind of a bet hedging strategy where they're putting some of their effort into trying to attract pollinators. Some of their effort is going into just asexual reproduction. The other thing, I guess I know some of the, Cuscuta, for example, has a range of different showiness in their flowers. Sometimes the flowers are very showy, sometimes they're less showy, or have more or less, nectar rewards. Other cases, you have, wind pollination as well. So yeah, sorry I don't have a more concrete answer, but I think that would be something that would be really interesting to just keep watching and reporting and
Sounds like some new area of research.
Yeah, new research for sure.
Lynn Michael asked, do host plants also grow toward the parasite through chemical signals?
So that's a really good question. So I think I mentioned the mycoheterotrophs will use chemical signals to recruit the fungi, which is their effective host. In terms of the hostorial parasites, I don't think any of the ones in Arkansas, will manipulate their host in that way. But I could be wrong too. In some groups of plants, you do see kind of like certain types of fungi, will cause behavioral changes in the things that they're infecting. I think there are some types of parasitic plants that will do that. I'm not aware of that happening around here.
All right. Jim Sullivan asks, is there Conopholis species in Arkansas?
I don't think there is actually. When I was checking over all the different lineages of parasites in Arkansas, I expected to see Conopholis here, the common Eastern North American species. But it looks like it only gets as far as like kind of Western Tennessee, Alabama. It kind of peters out before it hits the Mississippi River this far south.
I'm looking here at its range map on BONAP, and that's what I'm seeing. I'm trying to think, is there something else? I mean, it has its common name is, I believe, what, cancer root?
Yeah, that one. Yeah.
Does it happen to flower in May?
Depending on the latitude, yeah.
Okay, I'm just trying to think of, there was a species that I saw at Devil's Eyebrow Natural Area when I was out with the Native Plant Society. Actually, my very first hike with the Native Plant Society. This was back in, I can't remember what year now. It was the day after that natural area opened to the public. And we spotted it, or not, I don't know if it was Conopholis, but it was something with cancer in the common name. So maybe it was a different species. But that was one that I remember Theo Witzel commenting that it was, hadn't been recorded there in the natural area yet. But I mean, like I said, it could be, something completely different, but I do recall it being a chlorophyllous plant, one without chlorophyll. Do you happen to know if there was maybe another option, something else that we saw, or?
Yeah, so I think Brent posted in the chat that was Afilon uniflorum, and I know cancer is also a name for the Afilon.
Brent Baker was there on that hike. I remember that now.
This guy here. So yeah, sometimes these common names get kind of recycled.
Right.
A lot of the Orobanchaceae, or a lot of the Afilon also are called broom rapes.
Okay.
It comes from some of the European species that parasitize broom, the type of shrub. I'm not a super huge fan of that common name, but
yeah.
sometimes people hear that as well.
So what is another common name for the Afilon uniflorum that has cancer? And is it cancer weed or cancer? Does it have cancer?
Cancer root.
Cancer root
been used as well. Yeah.
Okay, it's just the same common name, different genus completely.
Yeah.
Gotcha.
But they're both holoparasitic Orobanchaceae, so they are closely related genera.
Okay. Gotcha. Well, I think that's all the questions we have. Really appreciate this presentation. Really great, very unique topic that we haven't been able to cover yet. So I really appreciate you sharing your knowledge and expertise and your time with us, Adam. This is really interesting stuff. So, and I want to thank everyone else for being here as well. Again, this recording will be put onto our YouTube channel. I posted a link into the chat box. So give a quick plug for our next webinar. It's going to be Wednesday, August 4th at 1 p.m. That's going to be by Dr. Subedi on oak diversity in Arkansas and biogeography. You can find out more about the Arkansas Native Plant Society on our website, anps.org. Check us out on Facebook at facebook.com/Arkansas Native Plant Society. I want to encourage you to join, and you can do that through a PayPal account on our website by going to amps.org/join. Adam, thank you very much. I really appreciate it, and looking forward to hopefully having you back to do another presentation sometime.
Thank you so much.
At one of our meetings.
I also just threw my email in the chat box, so if anyone wants to get a hold of me afterwards,
yes.
reach out.
Yep, and just to state it here, it is your last name, Schneider at hendrix.edu.
Yep.
All right, thank you all very much. See you soon.